presentation, many include these 4 parts. The work many include the first is a 3D assembly of the fully interconnected graphene carbon nanotube. We call this kind of hybrid scaffold as a GCNT web. Then we use this GCNT web to fabricate a 3D Cortex-Like network. And then we use this Cortex network to build a 3D Glioma infiltration model. As we see, the brain is formed by a complex assembly of cellular networks, where the neurons are embedded in an external matrix consisting of an intricate 3D mesh of proteins. The proteins can provide complex chemical, electrical, and mechanical signaling. Given this complexity, as well as the limitation of in vivo studies, it is important to develop in vitro models which could able to replicate the brain connectivity at various levels, and then ultimately provide a mimic of the human brain which is suitable for clinical applications. To construct a more physiologically relevant microenvironment in vitro, 3D culture system, especially material-based 3D assembly of neurons, have been developed for overcoming the limitations of 2D cell cultures. Therefore, when the researchers design the scaffold, they should first think about the biocompatibility. The scaffold should allow the 3D growth and adhesion of cells. Moreover, they should also provide the physical and chemical regulation for the cells and the communication between the cells and the cells, and also the communication between the cells and the ECM. So a lot of materials has been used to construct the neural scaffolds. The first is the natural materials, which we can the collagens, the matrix gel. However, the natural materials usually present lower mechanical stability. Therefore, polymers and some inorganic materials, especially graphene and carbon nanotubes, usually they have outstanding physical properties, and they have been widely used in neural scaffold applications. Here we have 2 samples, examples of the graphene's application in the neurobiology. One on the left, the graphene oxide, the graphene oxide form, they can promote the outgrowth of neuron fibers. And on the right, the 3D graphene form, they can promote the differentiation of neurons themselves. On the other hand, carbon nanotubes, they have unique one-dimensional geometrical structure, and also they have the wholly structure and good electrical support on intensive interconnection with and among cells. The connection can boost the electrical signaling, and they can modulate neuron growth and guide the functional reconnection of segregated spherical slices. However, also because of its one-dimensional structure, it is really difficult to form a real 3D interconnected structure of the carbon nanotubes. Therefore, a fully 3D geometrically electrically and mechanically connected carbon nanotube is required for biological study. So, when we investigate the literature, we can see that most of the studies, they cannot form a fully interconnected carbon nanotube structure. So the aim of our work is to construct a fully interconnected carbon scaffold. So the scaffold meaning combine the graphene and the carbon nanotubes. Here we have a chemical vapor deposition fabrication process of the scaffolds. Here we use the iron chloride confined graphite as the catalyst, and then we put into as prepared graphene form. Then we use the second step, chemical vapor deposition, to deposit the carbon nanotubes inside the holes of the graphene form. Here with the SEM, we can see that the legal form, and after the growth of the graphene form, and the last step, the carbon nanotubes can fill into the holes of the graphene form. And from the photo images, we can see that the colors with the growth of the graphene and the carbon nanotubes, the color become deeper. And also the SEM images, we can see that the thickness, the microstructure, the macrostructure of the scaffolds, they can reach the thickness of 1.5 millimeters. And with the amplified the SEM structure, SEM pictures, we can see that the carbon nanotubes fell into the holes of the graphene form, and they leave the space between the carbon nanotubes. And this kind of space has a very large distribution of the size. The size can range from hundreds of nanometers to tens of micrometers. And from the Raman spectra, with the D-bands, we can see that we have growth the carbon nanotubes inside on the graphene form, and also with the compressed and the electrical test, the GCNT web has enhanced the mechanical and the electrical properties than the usual graphene form. Then, after successfully prepare this kind of scaffolds, we culture the primary cortical neurons inside the scaffolds. We can say that after 8 days of culture, we use the map tube to mark the mature neurons, and with the SME 312, we can mark the axons. And then we compare the GCNT web and the normal graphene form. And we can see that we have a really denser neural network inside the GCNT web than the graphene form. Then also with the study of the actins, we can also have similar structures. We can have the similar observations. On the top are the GCNT web with the cells. The actin can mark all the cells. And on the graphene form, also with the SEM, we can see the cell body of the neurons and the glial cells. And then we compare the cell density and the cell volume. We found that in the GCNT web, we have a much denser cells. Here, to make sure that all the cells are inside the whole structure. So we check the cross section region of the immuno and the SEM. So on the cross section, we can see all the cells and all the neurons can fill the whole structure, and the thickness of the cells is more than 300 micrometers. Then to investigate if the neural network have functional electrical function activity, so we use the cathode image. We use fluoro to mark to test the cathode image, and here we compare the typical calcium transients between the glial cells and the neurons. So with the shape of the transient, we can distinguish the glial and the neurons. Here we also observed that we can also see the calcium transient from the soma and the process. So then we only focus on the neurons, on the neural networks. On the top and on the down, we compare the neural network inside the GCNT web and inside the graphene form. And here we plot the calcium transient. We can see from the picture, we can see they are different. Then we give quantify also here. Before the quantification, we also plot here the Rust plots and the cross correlation matrix. So here the Rust plots means every dot and every time. Here we have a spike of the causal image. So here we can see on 8 days and on 15 days of the culture, we can see the neural network inside the graphene form and the graphene and the GCNT web. They have different types. Also, compare the cross correlation. From the cross correlation, we can compare the synchronism of the neural network. From the GCNT web, we can see on 8 days the synchronizers are more significant than the 15 days. Here, from the quantification, we can also check. You see, here we compare the frequency of the custom transcends. At the beginning days of the culture, the frequency is very low, and from the 6 days, the frequency of the custom transcends begin to has a higher level. And even after 15 days of the culture, they have even stronger frequency. And then from the mean cross coefficient, we can see that at 8 days, the synchronism on the GCNT web is much higher than on the graphene form. So which means that in our GCNT web, we have a really denser neural network, and also we can observe higher synchronism of the neural network activity. Here we can have a comparison of the causal image. You see, on the left is the GCNT web. The neural network has a higher frequency and has a higher synchronism of the activity. Here we can see the difference. Then to investigate the neural network's application, we would like to investigate is to build a Glioma infiltration model. The ECM plays a really important role in the Glioma's infiltration. So here we also stain the ECM. Here we stain the aggregate and the laminin. Also here in the GCNT web, we can find a really denser ECM than inside the graphene form. Then we try to build a Glioma infiltration model. At the beginning, we capture the Glioma. The U87 is U87 is a Glioma cell line. So we first only capture the U87 inside the purified GCNT web without the neurons. And with the live cell image, we can see that the Glioma could move along the carbon nanotubes, and also during their movements, the shape are changing. The shape of the Glioma are changing. So then we do the quantification of the movement of the U87. Here we can recover the trajectory of the movement of a single cell here on the left. Then in the middle, we can recover the trajectory of the cells in both temporary and spatially, which means that we can see with the time change, with the time moving, the cells the position, and also on the spatial distribution, we can see the cells different. Then we can compare the single movements on X, Y, and Z and the different directions. Here we can see that on the X and the Y and the plane, the cells move much more than on the Z direction. We think this could be the gravity of the cells. So the cells move much more on the XY parallel more than on the Z direction. So after we have this technology to analyze the movement of the cells, then we try to do the co-culture of the neurons and the U87. So here, we first culture our cortical neurons for 8 days. Then we put the U87 into the culture system. Then we have the co-culture system. Here, the blue are the health neurons and the glial cells. The red are the U87. And from the left cell image, we can see that the U87 has a really strong communication with the health cells. And also with time change, then the U87 move along the carbon nanotubes with the shape change. So we compare the velocity of the U87's movement. We can see that on the cover slip, on the GCNT web without the health neurons, and on the GCNT web with neurons, the movement of the velocity of U87's movement decrease. So in this case, our model closer to the in vivo condition, because usually if the glioma's movement like the cover slip, I think the human will die maybe in some hours. So we decrease the velocity of the glioma's movement, the model closer to the in vivo condition. Then we recover the trajectory of the cells, and we compare the velocity, and we in every axis, and also we here is we try to test our model. We use the one drug. The drug's name is bilibistine. This kind of drug has been proved to block the motion of the glioma. So it can block the motion of U87. So then we use this drug to check if it works in our model. Here we recover the trajectory of the cells. On the top are the GCNT web without health neurons, and on the down the GCNT web with cortex with the neurons. Then we compare the cells movements before adding the drug and post adding the drug, and we can see that after we use the drug, the cell move much lesser, and they decrease. They decrease the velocity, and then they decrease the movement. Here we plot the velocity, and the drug works on the cover slip. The drug works on the single GCNT web, and also the drug works in our model. So in this case, our model is closer to the in vivo condition, and it worked to work as a glioma infiltration model. And this work has been published as an inside cover in Advanced Material. And also Wiley has has built a video abstract for us. And we can have a brief look. So in this work, we have firstly successfully chemical vaporization preparing of a fully 3D interconnected graphene carbon nanotubes. Then we use this GCNT web has built a functional 3D Cortex-Like neural network. And then we use this Cortex-Like network to build a glioma infiltration model. And this model works perfectly.